This video covers the IGCSE Biology Objective 7.4.7a on the 2025 CAIE syllabus to describe the digestion of protein by pepsin. So what is pepsin? Well, pepsin is an enzyme in the group of digestive protease. So in the previous videos, we've talked about that proteins are going to be digested by protease, and there is a gastric protease and a pancreatic protease. So now we know the name of the gastric protease, that is pepsin, and it is the enzyme that is released by the chief cells in the stomach. So let's take a look at what actually happens in chemical digestion in the stomach. So let's remind ourselves that the gastric juice is made up of three components: hydrochloric acid, mucus, and pepsin. Remember, the mucus is going to protect the stomach structure from the acid and the enzyme. And the acid, in the case of chemical digestion, is going to denature the protein of the food. So remember, proteins or polypeptides are folded up chemical structures. We talked about this in the section on biological molecules of protein. And the hydrochloric acid is going to merely just denature the protein, that is, change its shape. So it is no longer folded, it's now going to be stretched out a bit as depicted here in the diagram. It is then that pepsin is going to be able to do its work. So let's take a look at what that pepsin enzyme action is. So this enzyme is going to be able to break these proteins, the polypeptide chains, into peptide chains, that is, smaller sections of chains of amino acids. So we're looking at the first chemical reaction here. And so this is what happens in the stomach. So you can see here that the protein is not 100% digested into soluble molecules yet. So how does this process happen? Well, the process of protease digestion happens through a process called hydrolysis, and this is a chemical reaction. And this chemical reaction is going to break down chemical bonds due to a reaction with water, therefore the term hydrolysis. And you can see here in the diagram, we have two structures, and a water molecule along with the proteases are going to break those molecules at a certain point. One of the chemical bonds will break, and then we will have two chemical bonds. So the water molecule is in fact going to split. Each side of the split is going to attach itself to either end of that portion where the chemical bond used to be. So on the left side, the O and 1H from the water attaches to the left side of the chemical bond portion, and now that solo hydrogen attaches to the other side of the chemical bond that has been broken. And so now in this instance, we end up with two separate molecules that have been broken.